Joshua Lee, University of Alberta, Canada.
If you're anything like me, then you probably know when it comes to text messaging, autocorrect can be your best friend. Or your worst nightmare. Well, right now, inside every one of the trillions of cells in your bodies, there are genetic text messages being sent and received. And your body's genetic autocorrect is doing its best to keep up, but it's not perfect, it can make mistakes. These are called genetic mutations. They can cause genetic disease, and when it comes to genetic disease, there's no pill, there's no vaccine, there is no cure. That is the wall of genetic medicine. So, how do we break down that wall? Exon skipping is a molecular technology that uses small synthetic DNA-like molecules. Think of them like DNA band-aids that can stick to a region of a gene, say a region that has a genetic mutation. So then when that gene sequence is being read to make a protein and it comes to the place that's bound to the DNA band-aid, it skips over it, and that mutated region is not included in the final protein product. That's how you could treat a genetic disease. One of the most common and lethal genetic diseases in children is Duchenne muscular dystrophy. Characterized by progressive muscle wasting and muscle weakness, these children are wheelchair-bound by 10 or 12, on a ventilator not long after, and they don't live long into their 20s, and there's no cure. But exon skipping could help, because the majority of these patients have a mutation in a very specific region of a gene that prevents them from making an essential muscle protein. So if we can design these DNA band-aids to target that region, skip over it, and make that protein, we can treat the majority of those patients. Current clinical approaches using DNA band-aids are limited because they're only able to focus on a very small portion of that genetic region and are only applicable to about 13% of patients. At the University of Alberta, I have designed DNA band-aids that are able to target the entirety of that commonly mutated region in the gene responsible for this disease, an approach that's applicable to 45% of all patients. And using cells from these patients, I've successfully restored that missing protein. Now, what do you care about exon skipping and DNA band-aids? Well, who do you know who has muscular dystrophy? Who do you know who has Huntington's disease, or ALS, or cancer? This same technology one day could be applied to all of these genetic diseases and others, and that day is not far distant. So, in conclusion, thanks to new technologies like exon skipping, the wall of genetic medicine may soon come crumbling down. Thank you.